In the previous chapter, that is chapter 17, we did a hypothesis test for a matched pair problem. Now, if you remember, the matched pair problem was a single sample. There was just one sample, which is a group of individuals, and we took two different measurements from the same set of individuals. For example, if we are testing if a certain medication for blood pressure reduction actually works, then we take the before readings of blood pressure and the after readings of blood pressure. So same sample, two different readings. So we have two columns. One is the before readings and one is the after readings. And then the important thing to notice here is that both the measurements are coming from the same individual. So individual 1, we measure the before blood pressure and then the after. Individual 2, we do the same thing, before and after. The important thing to notice here is that both the readings, the before and the after, are coming from the same person. Now, in this chapter, we're going to look at problems that are related to two different groups of individuals. So which means we have two samples, sample 1 and sample 2. So, and both the samples contain two different sets of individuals, and we measure something. For example, if we are measuring the same thing, which is blood pressure, so we measure the blood pressure of group 1 individuals and we take the blood pressure readings of the individuals in group 2. But notice that they are two separate samples. We do get two columns of data, one coming from sample 1 and the other coming from sample 2. So that is the big difference between a two-sample problem, which is two different samples, two independent samples, giving us certain readings, let's say of the same thing, which is blood pressure. But the difference is that we have two columns of data and each one is coming from a separate sample. So that's the big difference that we need to notice when we're reading a problem. Is it a matched pair problem, which means is it coming from a single sample, or is it a two-sample problem or a two independent sample problem, in which case is it coming from two separate samples, two separate sets of individuals. In the case of a matched pair problem, if you remember, we created a new variable, x. We called it x. And we took the difference between the two measurements, a and b, for example. So we did either a minus b or b minus a. And the difference between the two variables that came from the same set of individuals was called x. And what we did really was converted it into a single sample problem. And then we went about doing the hypothesis test as if it were a single sample problem. And the single sample was x, the difference between the two measurements. In the previous case, in the matched pair problem, it made sense to take the difference between the after readings and the before readings, for example. It made sense because it was coming from the same individual. It makes sense to say, did the medication reduce the blood pressure in this particular individual? By what quantity did it reduce the blood pressure? And that's the difference, and that we called x. Now, in the case of a two-sample problem, it doesn't make sense to take the difference between these two numbers because they are coming from two different individuals in two separate groups. So in this case, we have to deal with it as a two independent sample problem, and we cannot take the difference and convert it to a single sample problem. Now let's think about where the two samples are coming from. In certain cases, the two independent samples could be coming from the same population. And in some cases, it's possible that the two samples are coming from two separate populations. So, for example, men versus women, or it could just be two different sets of people, maybe two separate groups of women. So, either way, it is a two independent sample problem. Now, this is definitely not a matched pair problem because we have two separate independent samples. When we read a question, it is important to be able to identify the type of problem. That is it a single sample problem? Is it a matched pair problem that can be converted to a single sample problem? Or is it a two independent sample problem? So let's look at a few examples to see if we can identify the type of problem, which is the first step to solving problems related to inference in statistics. So let's look at the first example. We have 24 hydrological stations, so that looks like our sample, and our sample size is 24. And what are we measuring in each of these stations? We are measuring the acidity of rainwater, and it is being measured in two separate years. So in 1995 and 2005. And what we want to do is estimate the average change. So the minute we see the word change, we know we have to take the difference between two readings. So this looks like we have two columns of data, one from 1995 and the other from 2005. But reading the question carefully tells us that the same hydrological station is giving us these two numbers, one number in 1995 and the other number in 2005. We have the second hydrological station, we do the same thing, one number in 1995, another number in 2005. So we have to stop and think about how these quantities are being measured. Now clearly there's just one sample and the sample size is 24, and we are taking two measurements at two different points in time. So clearly this is a matched pair problem. So if it's a matched pair problem, then what we do is take x, which is the difference between the two measurements, and that becomes our new variable. So we convert a matched pair problem to a single sample problem and complete the construction of a confidence interval in this case, because we are estimating the mean. So we estimate the mean using a range of values, and that is called a confidence interval. Now in the second problem, the researcher is studying how synthetic fabric decays in landfills. So as an experiment, he buries 10 strips of polyester fabric in well-drained soil in summer. So he buries these 10 strips. And what he's going to measure is the breaking strength of these strips after a period of time. So 5 of these strips are removed or dug up after 2 weeks, and their strength, breaking strength, is measured. So there's a set of 5, 1, 2, 3, 4, 5 fabric or strips of polyester fabric, and their breaking strength is measured after 2 weeks, and then another set of 5 are dug up after 16 weeks, and again their breaking strength is measured for each strip. So here clearly we have two separate samples, one that is dug up after 2 weeks and the other that is dug up after 16 weeks. And each time we are measuring the same thing, which is the breaking strength. But as we are reading the problem, it should be clear that they are two independent samples, and we treat this as a two independent sample problem. So in the next problem, a chemist is testing a new analytical method for finding or measuring concentrations of given specimens. So what she does is she takes a specimen with known concentration, and then she takes 20 separate measurements of concentration using this new method. So here clearly there's just one sample of size 20. So these are the 20 measurements. So it's a single sample problem. There are no two samples here. So clearly this is a single sample problem. So these three examples kind of show us how to read the problem and decide what kind of method to use. Is it a single sample problem? Is it a matched pair problem? Or is it a two independent sample problem? The question will clearly explain what kind of a problem it is. We just have to read it carefully and identify it. When we use the two sample procedures, then there are a few conditions we need to check before we use the two sample or treat it like a two sample problem. The first thing is, are the two samples independent of each other? Are they coming from either two distinct populations or two different independent samples from the same population? Making sure that the populations from which we are extracting the simple random samples are normally distributed. So the teach sample procedures, we have two separate sampling distributions, one for each sample. So the formulas that we use change accordingly. If we have a two sample problem and we need to construct a confidence interval, then this is the formula that we use. Notice that we have numbers x1 bar and x2 bar. So x bar in general is the mean of the sample, so it's the sample mean. We have subscripts x1 and x2 for the two separate samples, which is sample 1 and sample 2. So x1 bar is the average of sample 1 and x2 bar comes from the average of sample 2. We also have S1 and S2. In general, S is the sample standard deviation that comes from the sample, and the two subscript S1 and S2 tell us which sample we are referring to. So the S1 would be the standard deviation of the first sample and S2 would be the standard deviation of the second sample. Now clearly N1 and N2, N is the general letter that we use for sample size, which counts the number of individuals in the sample. The subscript 1 is for the first sample. So N1 is the sample size of the first sample, N2 is the sample size for the second sample. N1 and N2 do not have to be the same. It's possible that the two independent samples are of different sizes, so they don't necessarily have to be the same. Now, if we were doing a hypothesis test for a two sample problem, then this is how we calculate the t statistic, which eventually gives us the p-value. So this is the formula that we use. Again, notice x1 bar minus x2 bar. So these are the means of the two separate samples. S1 and S2 are squared in this formula, and S1 is the standard deviation of sample 1, S2 is the standard deviation of sample 2. And again we use the sample size N1 and N2. N1 is the sample size of the first sample and N2 is the sample size of the second sample. We plug all the numbers in to get our t statistic. We take the t statistic to the table C, just like we did before. So this goes to table C, and from there we find the p-values to conclude the hypothesis test. The t star here, clearly we've used this in the previous chapter. Again, t star comes from table C, which is the table for the t distribution. Now, one last thing to point out in this two sample procedure is if you're going to use table C, we need to know what the degrees of freedom are. How many degrees of freedom does this problem have? Previously, we never had to think about it because there was just one sample of size n. We subtracted 1 from it and we got the degrees of freedom. Now, in this case, we have two samples and each of a different size, let's say n1 and n2. Now, the degrees of freedom would be the smaller of the two. So here we have n1 minus 1 for this particular sample, degrees of freedom n2 minus 1 for the second sample. Whichever one is the smaller, and that's what we need to remember, pick the smaller of the 2 degrees of freedom, and that is what we would use to figure out the p-value for this particular t-statistic or the t-star for the confidence interval. Smaller degrees of freedom. That's the important thing since we have two different sample sizes.